Hello, everybody. Very happy to be here today with you for another session of Expand Your Horizons. We already have hundreds of people joining, so thank you very much. Now, without further ado, hello, Anne. How are you? You're in mute.
I just said good morning, but it's good morning here. It's probably good afternoon in a lot of places.
Indeed. Thank you very much for being with us, Dr. Anne Simon, the founder and CTO of Silvec Biologics. Before we give you the stage, if it's okay with you, I'll take a few minutes to set the context for our conversation today.
Absolutely.
Good. Let me start with a very, very bold statement. In 10 years, every citrus tree on Earth will be dead. So very, very scary statement. And in fact, it's not only citrus. There are other diseases that affect other trees like olives and vineyards and apple and nuts. And for citrus, people have heard about the pandemic called HLB or greening, especially our nanofilm guys that are focusing on agriculture, but not many people are aware of this pandemic that Anne will talk to us about in a minute. Can you imagine a world without citrus or oranges that will cost $20 or $30 each because they will have to be grown in a protected greenhouses? So this is one scary thought I would imagine. Now, what is actually happening with those diseases are these bacteria that inhabit the veins of the plants. And what happens is that it's very difficult to heal and address those diseases because of the nature of how plants carry substance in their veins. So what Silvec, the company that Anne founded, is targeting is to address those issues. The other important thing is to mention is that the big issue is not with the bacteria itself, but the autoimmune reaction of the tree to that bacteria. So in essence, the tree kills itself more than the bacteria kills the tree. And again, we'll hear about it by Anne. The novelty of what Anne and her team developed is to use kind of like viruses as a vehicle for antibacterial agents. And this is, a real breakthrough. We got so excited with Silvec's breakthrough that we decided to invest in the company through Orbia Ventures. So before I switch to Anne, I just want to introduce you to the person that is joining us today. So Anne received her BA from the University of California. She has a PhD from the University of Indiana. She was a professor in University of Massachusetts and has been in the Department of Cell Biology and Molecular Genetics at the University of Maryland. She is also the head of the University of Maryland Virology program, and she teaches the introduction major biology course that is receiving the highest awards by the students in that university. Her research that led to Silvec Biologics is focusing on RNA viruses. She is also the recipient of the Franke Prize for research on plant virology. And for the past 14 years, she is a senior editor in the Journal of Virology, which is the top international journal in the domain. Maybe two fun facts that you might not necessarily find on Google if you search for Anne. The first one is that for the 11 seasons of X-Files, Anne was the science advisor for the series, helping them make sure that what they invent, makes sense. And consequently, she also wrote the book, The Real Science Behind the X-Files, that you can buy. And the second piece of interesting piece of information is that Anne is an enthusiastic equestrian and dressage rider. And I had to look it up because I had no idea what it means. It basically means that you are a professional horse rider in different forms and formats. And you actually have your own Andalusian horse called Tormento, right?
I have two. Yes, Tormento was one.
Nice.
I did not name him that, but he's kind of like that.
Good. So, Anne, the floor is yours. Share with us the amazing story of your research that led to Silvec, and then we'll keep some time at the end for Q&A.
Thank you very much for that wonderful introduction. It's really great to be here today. I haven't ever given a talk like this before, but I hope you'll find it interesting. I'm going to tell you today about a very unexpected journey, and you'll see why it was so unexpected, I hope, as we go on. So I was told to start a little bit with my history. And so as a budding scientist, not just me, but anybody, there's a lot of very important decisions that you have to make. So about 30 years ago, which was after my undergraduate years, my graduate years, and all my postdocing years, I had to decide between, do you go into industry or do you go into academia? And there are reasons for choosing one over the other. For industry, well, there's no teaching, although I like teaching, but it takes time away from research. There's no grant writing, that's a big plus, and there's more money. For academia, on the other hand, there's teaching, there's grant writing, there's less money, but there's also freedom. The freedom to choose what you want to study and the freedom to change your mind and go after something that comes up that's interesting. So I ended up choosing academia because I liked the idea of freedom in my research. So then the second decision is, because I knew I wanted to work on viruses, do you work on model viruses or important viruses? So what's a model virus? A model virus is, you choose it because it's so simple, it's tiny, it's easy to work with. And when you work with model viruses, you can make a lot of fundamental advancements to the field of virology. Then there are important viruses like today's SARS. These are viruses which you might say, you work on to benefit humanity. So I wasn't back then really, that interested in benefiting humanity. I was interested in working on simple viruses. So I chose to study model viruses. And then the next thing is, what kind of virus do you choose? A virus that infects animals and humans, or a virus that infects plants? And this was actually a very simple decision, even though my background was not in plants. Plant viruses are the simplest viruses. And once a virus gets inside a cell, they all have to do the same thing. They all have to reproduce, and they have to get out and infect other cells. So I chose to work on plant viruses. So here's one virus called turnip crinkle virus. And to show you just how unimportant it is, it's only been found 3 times in the history of the planet. So this is a pretty typical virus. It's an RNA virus. It encodes, which means it's a blueprint for 5 proteins. So the ones in purple here make the enzyme that reproduces the virus. These 2 proteins are involved in movement, and I'll talk about that in just a little bit. And then this one is called the coprotein. And from the coprotein, you get a virion. And you're probably used to seeing this now. This is, of course, the virion for the SARS coronavirus. This is not to size. This is a lot bigger. And SARS, of course, is a very important virus. And like so many important viruses, it's also a very complicated virus. So just to compare the SARS virus with the virus that I study, this has 5 proteins that it specifies. This virus is not 4,000 nucleotides, which make up its RNA. This is also an RNA virus. It's 30,000. And instead of 5 proteins, it's 29 proteins. So it's hugely complicated. And of course, I don't choose to work on things that are that complicated. So for a large number of years, I worked at University of Massachusetts and then University of Maryland. And more recently, I chose to work on a different virus. These are called Umbra viruses, and these are even more simple than this one of the world's simplest viruses. And so this is a virus called PE Nation mosaic virus, and it really doesn't harm pea anywhere. But it's got the replication proteins. And then these two proteins are very important for movement of the virus around the plant. Notice that it's missing the coprotein. So it doesn't actually specify its own coprotein. It's always found in plants with another virus, a second virus. And that other virus provides a coprotein for this one. So this is even simpler than this. How a virus moves in the plant is very, very important. And fortunately, plant viruses have very limited hosts. So a plant virus doesn't infect all plants, just like a human virus, infects humans, but there are viruses that maybe your dog has or your cat or your horse gets, but you don't catch those viruses. And in plants, what decides whether a plant is a host for a virus is whether or not it can interact with these movement proteins. So movement proteins on a plant virus allow the virus to be able to get through these little passageways between cells. And what's guarding these passageways is another protein. And you have to be able to have an interaction here between the protein that the virus produces and the host protein. And if you have an interaction, then the virus can infect. However, if there is no interaction, if you can't, kind of fit together like a key in a lock here, then the virus cannot infect. So these movement proteins decide whether or not a plant will be a host for your virus. So one day as I was searching through the database, and everyone that sequences viruses puts their sequence in this database, the NCBI database. So all the SARS viruses that are being sequenced, all those sequences end up in the database. What I found in the database was this. And this was really shocking to me because it looks like this Umbra virus right here. In fact, these proteins are related, but it's missing something. It's missing movement proteins. This is not present. So obviously it would need a helper virus. But I was very curious about this, and I found out a little bit about it because no one was actually studying it, but it had been put in the database about 5 years previous. And it turned out that it's only been found once in the history of the planet. And it was found in 4 limequat trees in the 1950s. So that's a lime crossed with a kumquat, gives you a limequat, which I guess you can eat, maybe. I've never actually seen one. But it turns out that at the time, they didn't know what was in these plants, these limequat trees, because all they saw as far as disease was this, just these little tiny yellow lines on the leaves. And it turns out that you can take a piece of an infected tree and graft it onto, virtually any other citrus, and you could get the disease transferring with it, although you only get symptoms on lemon and lime. So I was very curious about this, and I wanted to study it. I didn't think actually when we started studying it that we could even get it to replicate in a single cell. And that's because this protein right here in yellow on top has a couple different functions. In fact, when you're a really simple virus, all of your proteins have to do multiple duties. So this protein right here does a second duty. It not only is involved in movement, but it is also involved in protecting this virus against the cell's defenses. And if you don't have this protein present, you don't get any replication. This thing is immediately degraded in the cell. So since it didn't have this protein, I thought, well, it's not even going to replicate. So we decided to try to see if it would replicate all by itself in a cell. And the answer turned out to be yes. So I was very surprised by that. And then the graduate student who was doing this work came to me and said, I want to see whether or not it can move all by itself. But I told her, well, the answer is going to be no, because all plant viruses have movement proteins. They have to have movement proteins, and this doesn't. So obviously it can't move. So she did the experiment. She tried 10 plants to see if it would move. And in one of the plants, it moved all by itself, which was impossible, but clearly not impossible. So this was really astonishing to us because it shouldn't be able to move. So we asked the question, how is it moving without movement proteins? And it turned out to be really interesting. It's using a plant protein to move. So instead of specifying a movement protein, it's using something that's already in the plant. And this thing attaches itself to it, and it's able to move it around. And of course, the plant protein, which is also moving the plant's own RNAs around in the plant, it automatically has to have its partner here. So there's no problem with this thing actually being able then to move. And then that led to a very interesting idea. If it's using a plant protein to help it move and the partner is always there, and this protein is in every single plant, it's very well conserved, then shouldn't this little RNA be able to infect every plant? So we looked around to see, has anyone else found anything like this that's infecting other plants? And the answer was yes. It had been found in cactus and corn and fig trees, sugarcane, strawberry, and the strawberry, we just found that this weekend. We've been able to put it in tomato, tobacco, and cucumber. So it looks like it's able to infect every plant. This is unheard of. So what do you do when you find something like this? And when I used to, give advice to the X-Files, one of the questions that Chris Carter asked me was, if you were a scientist and someone brought you an organism that, clearly was not from this planet, who would you call? And I said, well, I, call the government and tell them that there's something here that's alien. Well, this isn't alien, but I did end up calling someone. And the first person that I called was my brother, Raphael. So to understand this story, you have to meet my brother. This is actually a current picture of my brother. So you can see he's a couple years younger than me. He got his PhD in Berkeley in engineering. So he didn't know anything about biology. He spent 3 years as a consultant for McKinsey, and he became one of the world's authorities on water purification. Didn't have much to do with plants, obviously. He spent 8 years as the chief operating officer of a company called Xenon Environmental. And he took it from $50 million to $750 million during this time when it was bought out by GE. And he was named the top chief operating officer in all of Canada, this is a Canadian company, by Canada's leading business magazine. So after he left Xenon, when it was bought by GE, he then spent 7 years as a venture partner in a company called Vantage Point, a $5 billion company, specializing in water purification. And because he had me to, talk to him about stuff, in ag biotech. And then he was the president of his own consulting company as well. So every year when the family got together at Thanksgiving or at Passover for Seders, Raphael would always ask me the same thing. He'd say, when are you going to have something that I'm going to be interested in? When are you going to invent something or discover something that I'm going to be interested in? And I said to him every single year, Raphael, I will never have anything that you'll be interested in. I work on model viruses. I work on unimportant viruses. Nobody's interested in turnip crinkle and PE nation mosaic virus. But then I started to realize that there was something very interesting about this new little RNA, this citrus yellow vein associated virus, CYVAV. So I called him and I said, Raphael, I have something interesting, finally. And this is what I told him. I told him about these worldwide pandemics that were affecting trees and vines. And the one that most people might know of is the one that's affecting oak trees, because a lot of people have oak trees in their backyard and the oak trees are dying. And they're dying by the thousands in the US and in Europe. They're dying from diseases caused by bacterial and fungal pathogens. And the diseases are spread by us, spores on us, on clothing, things like that, and also by insects. And they're made much worse by climate change. Climate change is weakening the trees, and it's also affecting the spread of insects. And there's no treatment. There is no treatment or cure for any tree disease or disease of vines. So it's much worse than oak trees, as Shai told you in the introduction. You've got diseases that are affecting entire industries, like citrus greening, what we're going to talk about today, caused by a bacteria. And literally in 10 to 15 years, all citrus trees that are being grown outside of glassed-in greenhouses will be dead. Olive tree blight in Italy right now, killing millions of 1,000-year-old olive trees. I don't know right now if it's in Israel, but it's certainly going to be spreading. This bacteria that causes olive blight is the exact same bacteria that's decimating the grapevine industry, Pierce's disease. And this bacteria has 500 different host plants, crop plants that it infects. The entire grapevine industry in the US is currently threatened by this bacteria. And there's a lot of viruses. There's viruses that are infecting grapevine. There's viruses infecting citrus. This is one really terrible virus called citrus tristeza virus, which causes citrus quick decline. So you can figure out what it does to citrus trees. And it's killed 50 million trees. So here's an infected tree. Here's a normal tree. Here's grapevine that's being infected by Pierce's disease. And this is what olive groves are beginning to look like in Italy. And so there's one thing that all of these pathogens have in common. As Shai said, they all inhabit the veins. So the veins, it's the phloem of the plants. And the veins have two different places where they conduct sugars that feed the roots. The sugars are made in the leaves, and then they go down and they feed the roots. And then there's water transport. The bacteria that's causing Pierce's disease, this grapevine disease, and that's so infecting the olive trees, lives in here, in the xylem, which conducts water. The one that's infecting citrus greening and CTV, the virus, lives here. And it's virtually impossible to get at these places and try to get rid of these viruses and these bacterial and fungal pathogens. And so I'm going to tell you about citrus greening today, because this is the first thing that we're concentrating on. And it is considered the #1 agricultural problem facing the world. So as Shai said, I want you to imagine. So we're going to give you a chance to imagine just for a moment a world without citrus. So no orange juice or grapefruit juice. No, this is a particular favorite of mine, perfumes that are scented with citrus scents like lemon. Fish without lemon? Okay, not going to be as good. Cleaning products, these are just ones I have in my house. All of these use citrus scents. And then, of course, margaritas and mojitos. Say goodbye. So a world without citrus is going to be a very different world. And it's being caused by this pathogen, this bacteria called Liberibacter asiaticus. And people ask me all the time, well, when did this start and where did it start? So we have a publication that we found from the 1920s, which talk about the disease. They didn't know what it was, what was causing it, but they talk about the psyllids. And psyllids are tiny little insects. So here's a whole bunch of psyllids on just one leaf. And they talk about the disease in citrus in India and also in China. So it started sometime at the turn of the last century in India and China. And what it is it's caused by this bacteria, as I said, and the bacteria live inside the psyllids. So in the psyllids, this is a very beneficial bacteria. So I think most people know now that we have a lot of bacteria inside us, our microbiome. In fact, we have more bacterial cells in us than we actually have cells. And the bacteria, most of them, the vast majority of bacteria inside you are very beneficial. And the bacteria inside the psyllid is very beneficial. It needs this bacteria, but it doesn't have any way to transfer the bacteria to the baby psyllids, what are called these little nymphs. So the way it transfers the bacteria is it feeds on an uninfected tree, deposits the bacteria inside the sap in the phloem of the tree, lays the eggs. The bacteria put off a scent, very interesting, an odor. We can't smell it, but the little nymphs can. So even though there's almost no bacteria inside the tree, the little nymphs figure out where the bacteria is, suck it up, go off, fly away, and infect other trees. So meanwhile, the tree, as Shai said, if the tree would just accept that there's a few extra bacteria in it, we wouldn't have a problem. But the tree mounts this enormous, completely unnecessary defense against this tiny little amount of bacteria. And what happens is the tree makes these sugars, and it makes a lot of a particular protein, and it clogs up its veins with the sugar and the protein and kills itself within a few years. The trees are just not very bright. So where is this disease right now? Citrus greening, this is 2 years ago, and it's already out of date. It's all over China. It's all over India. It's all over Iran. It's all over Africa. It's in a lot more places in Africa right now. It's all over Brazil. In the US, in Florida, every tree is infected. 75% are already dead. Brazil's lost 58 million trees. It's all over the Caribbean. And it's moving into Spain. And it's moving very soon, they figure, into Australia. And you just can't keep it out. So what they're using to try to detect if a tree becomes infected is they use these dogs. So it takes a couple years before you actually see the symptoms of it. So the symptoms are this: you get this yellow coloring in the leaves because the sugars can't go through the veins, so they clog up in the leaves, which turns the leaves yellow. And the fruit becomes inedible after a few years. But the trees stay pretty healthy looking up until then. You can't even tell if they're infected unless you use these dogs. So this is really very clever. The dogs have been trained to smell the little scent that the bacteria put out. And what the dogs do is they release the dogs in the field, and the dogs will sit at a tree where it smells the scent. So right here, this tree is infected. And the only way you know it is with the dogs. The tree looks perfectly healthy. So when they released the dogs recently in Southern California, in the Riverside area, they didn't think they had any citrus greening there. The dogs sat at 75 trees. There were 75 infected trees there. And it's now moved up into the Los Angeles area. It's in the groves in Southern California, in Oxnard and Ventura. And the orange growers in California are just panicking right now because they know in a year or two it's going to be all over. So what are the treatments for citrus greening? Why aren't there treatments? USDA has spent $500 million on treatments, and other countries are spending a huge amount of money as well. So where has all this money gone? Well, what are they doing in Florida right now? They are spraying 650,000 pounds a year of medical grade antibiotics on the trees in Florida and in California. And it's the largest use of medically important antibiotics, spectinomycin, oxytetracycline. It's doing nothing. It's sitting there on top of the leaves. It does not get into the veins. And it's producing bacteria that are resistant to antibiotics. So if your country isn't doing this right now, it shouldn't start. This is a terrible thing to be doing. But unfortunately, our last president approved it, even though the EPA said no. So they're doing it right now. What else are they doing? Well, you can inject into trees antibacterial peptides, for example. But the problem is you have to keep injecting them in. And the cost of doing these injections and the antibacterial peptides is more than the cost of the tree. So it isn't worth it for a farmer to be doing this when the tree isn't worth as much as it's costing them to do it, the amount of oranges that they're getting. So they're doing this right now in Florida. They're covering all these trees with netting. The problem is it's causing the trees to be extremely hot underneath there. And also you have to lift the netting if you want to get any fruit. And then all the insects come in. Or they're making these giant screen houses in Florida where they screen them in and hope to keep the insects out. But of course, in a lot of places, you get very bad weather. And all you need is hail or a hurricane, and your screen house is going to let in all of the insects. So it's almost impossible to keep them out. So what's the answer to this? They've known about citrus greening now for many years. Why isn't there an answer? But we all know that there's an answer. There are actually two answers to it. And the answer is these small RNAs. And everybody in the field pretty much accepts it, that you can use tiny RNAs. You can develop these as therapeutics. They're being developed as therapeutics in humans against bacteria, and they can be used to target bacteria and viruses and fungi, nematodes, insects. You can make a GMO, a genetically modified tree that produces these small RNAs. And they did this in the papaya industry in Hawaii. They saved the entire papaya industry by targeting a papaya virus that was decimating all of the papaya plants. And now all of the papaya plants look just fine, and they keep the virus out. So it's very well known that siRNAs will work very, very well. And siRNAs, they're called small interfering RNAs, can also target tree gene expression. It can keep a tree from producing the sugars that clog up its veins and from producing all the extra protein that clogs the veins. So this is well known that if you made genetically modified orange trees, let's say, that are producing these siRNAs, you could control citrus greening on the tree. The problem is the growers don't want a GMO approach. There's nothing wrong with genetically modified organisms. It's perfectly safe to eat. But of course, you know that there's issues in Europe and in a lot of different places. And they're worried about the consumers. So the growers do not want a GMO approach. They want something else. And so what is the something else? Well, we also have known for a long time how to do a non-GMO approach. We can use viruses as vehicles to deliver these small RNAs. So how do you do that? So if you have an RNA virus, that's a virus right here, and you can add a sequence of the RNA that'll make an siRNA. And inside the plant knows how to do this. It cuts this out, and this is then your siRNA. So you can control gene expression. You can target viruses or other pathogens by simply adding these things to a virus and then infecting your tree. And if the virus spends its time in the veins, like a lot of viruses do, then the virus is in exactly the tissue that you need to be in to go after bacteria that's living in the veins. So we know this. So why aren't we doing it? Why hasn't it been done for like the last decade? Well, the problem is there are no good citrus viruses to do this with, because let's think about it. Where has all of the money gone to study plant viruses? It goes into studying really bad viruses that cause a lot of disease. You certainly don't want to use a virus that causes a lot of disease as a way to go after another problem in the plant. So there were no good citrus viruses to do this with until now, until I realized that this little virus-like, it's not really a virus, it's a virus-like RNA, infects citrus and doesn't cause disease. And that is what I told my brother on the phone. And Raphael got extremely excited. And after a weekend of trying to learn an entire field, which he did pretty well doing, he told me that it was the most exciting thing as a venture capitalist that he had ever heard. And he literally dropped almost everything that he was doing, all of this consulting work, and wanted to devote all of his time to developing CYVAV as a way to target citrus greening. So the lab was able to get about $1 million in grants, because I'm good at writing grants, from the National Science Foundation and the USDA. And we were able to show that this little RNA was actually a great way to deliver siRNAs. It could be done. And I must admit, it took every bit of knowledge gained over my 25-year career up to that point to make this happen, because it wasn't easy to do. There were lots of problems along the way. I would call Raphael up and tell him, and he'd say, it's an engineering problem, engineer it. And so I would take that and try to engineer it, and a lot of failure, but successes. And we were able to make it into a vehicle to deliver siRNAs. So one of the first things we did as a proof of principle, as we say, is we used it to deliver small RNAs to eliminate this very nasty virus in a laboratory plant. So this virus is called citrus tristeza virus. And what we did is we developed an siRNA that would very specifically target just that virus, nothing else. And so this is the experiment that we showed. So what we did here is we put the virus, this nasty virus, and this virus is carrying a protein, it's engineered, that makes it turn green. Okay, so where you're seeing green, that's where the nasty virus is. And we put in the nasty virus with the normal version of our CYVAV, our little RNA that doesn't carry any siRNA. So it's not going to do anything. And it doesn't do anything. There's plenty of virus here in the leaf that we put it in, and it goes throughout the plant and gives you a bad infection of this laboratory plant. But if we put it in with our virus vehicle carrying this little siRNA that targets citrus tristeza virus, none of the plants become infected. It completely wipes out this virus. So when we realized we could do this, University of Maryland patented the technology, and Silvec was founded and licensed the technology from the University of Maryland. So that brings us to Silvec. The goal is to develop the first universal platform for the treatment of trees and vines, because this little RNA of ours should be able to infect everything. So if we can develop it for citrus, we should be able to develop it for oak trees and grapevine and apple and all of these diseases that are out there. And industries right now are suffering $100 billion in damage yearly from pathogens. So the market is huge, and there's no other competing solutions besides injecting trees, which is takes a lot of manpower to do the injections and costs a lot of money. So the president of the company is Raphael, and recently Raphael gave me a promotion, as he said, to CEO, because it was the only way to get the tax credits in the state of Maryland for the company. Is the CEO had to be a resident in Maryland, and Raphael lives in Southern California. And for some reason, he doesn't want to come out here to all the snow and ice and seasons that we have out here in Maryland. He would rather enjoy Southern California right now. So Silvec right now Our doors opened in August 2020, and we have 5 PhD employees with extensive experience. We've hired like the A+ team. Everyone that we talked to got wildly enthusiastic. We had one person who was in his own startup company. He folded his startup company to come and work because he said the technology they were trying to develop to treat citrus just was not working, and it wasn't anywhere near as promising as what we had. So we're located in a place called Rockville, Maryland, which is a nice corridor for biotechnology. And I'm still continuing as well at the University of Maryland. They let us do this. And I have a lot of funding still to work on developing this as a platform and understanding its biology. So I have 4 postdoctoral scientists and 1 PhD graduate student, the one who did the original experiment. It was all from that's interesting to think. One of these 10 plants, it went throughout the entire plant. And based on that, everything we're doing now was based on that one result of that student who is on the patent, of course. And she'll be getting her PhD this year, and she'll be joining the company. So in the first 30 years, I was in academia working on model viruses because they're so simple, and of course, model plant viruses. And now in the next 5 years, I find myself working in industry as well on an important virus, something I never thought I'd be doing, still in plants. And using all of this knowledge that was gained working on model viruses to attack this problem, this really significant problem that's affecting citrus, grapevine, and so many other plants. So right now, the road from Silvec Biologic continues. And I think this is going to be such an exciting journey. And I'm so happy, really so happy that Orbia is going to be our partner in this. So with that, I'd like to thank you for listening. And I'm more than happy to answer any questions that you have.
And thank you so much. It was really exciting. It was eye-opening, and you managed to deliver highly technical and sophisticated information in a way that even I could understand. So thank you for that. Couple of questions that are coming through. We might not have time for all of them, but let's try. If everything goes well according to your plans, what should we be expecting in the next 2-3 years?
So right now, the biggest benchmark that we need to get through is putting this little infectious RNA back into citrus and getting it to infect these other plants. So we have about 6 different ways that we're trying right now to get it to reinfect citrus. And there's several ways that we know will work, but we're trying other things as well because it's simpler and it leads to fewer complications down the line. So when we get this back into citrus, we can almost immediately start testing to see if we can target the bacteria. So we know now, just in the last really 2 weeks, we know that we can use these small RNAs to very specifically target bacteria and the bacteria that we want to target too. So I think that what we're going to see is this approach with the engineered virus vector, the CYVAV vector, with citrus. And also what I want to say is one of the questions that we get asked is, well, what about organic growers? So we've talked to the organic growers and they say, well, we don't mind having this little virus inside. That should work. That should still be organic, but we don't want the little virus to be engineered. So could the little virus by itself help citrus? And the answer, tantalizingly, is yes. We really believe that just this thing by itself can help the citrus tree to overcome citrus greening. And the reason for that has to do with this plant protein that moves our little RNA around the plant. It turns out that plant protein is the same one that clogs the veins. So instead of clogging the veins, it'll be attached to our little RNA. And if it's not clogging the veins should stay open. So we're hoping that we can have an organic approach, a conventional approach, and we've recently started partnering with the entity in the U.S. that supplies all the grapevine to the entire United States and also a lot of other places as well. And so we're immediately going after grapevine to see whether or not we can target these bacteria. So I think that if everything goes well, we're going to find that we're going to be able to reduce HLB in mature trees. I think we might be able to do it. If the trees are too far gone, I don't think anything's going to work. But the idea is to vaccinate. We want to be able to vaccinate seedlings. We want to be able to put things in the ground that will not get infected by some of these terrible viruses and that will not get infected by the bacteria that causes HLB. That I think we can be successful in. So, and I think it's going to take us a few years, but if everything goes right, we're going to be able to control not only those diseases, but hopefully the disease in olive trees, and really go after a number of different trees. So we'd like to be able to, if we can get this into grapevine and into citrus, then really the floodgates open. Then the more people that we have, the more diseases we can approach now, as opposed to waiting until we have a success with like citrus greening. We know we can be successful against viruses. And so the question is, can we also be successful against bacteria? We would like to be, simultaneously working to control a number of diseases. So I hope that answers the question.
And related to that, and this comes obviously from being the largest irrigation company in the world, do you believe that for mature trees, irrigation and specifically drip irrigation could be a successful delivery mechanism through the roots?
So far, nothing that's absorbed through the roots has worked. So they've tried that. And why it's not successful through the roots, it might, I'm not sure, but it's not a way that viruses get in. So viruses always get in through wounds from insects and things like that. And we have never been, this little RNA of ours does go down into the roots. So another thing is that maybe people can come up with, I'm not really we call it plant people who work on plants are plant people. So I don't have a real background in plant physiology, but let's say that someone can come up with a way that roots can better absorb water. We could use this little RNA to change the gene expression in the roots so that they could absorb water better. Let's say there's a way to do that, to modify the xylem or something like that. So I think that this could be useful for drip irrigation, but actually as a way to get things in, it's an obvious route, and people tried it for many, many, many years and were not successful. So I don't think that that's the way of getting something in. But I think that we can always get things in by grafting. And that's the goal, is you make a mother tree. The mother tree has CYVAV, this little RNA with whatever little siRNAs. You can have multiple siRNAs too. So the mother tree has this in it. You take a little piece of it, and a grower can just then make a little graft. People are used to making grafts into mature trees. You make a little hole, you put the graft in, bandage it up, and it starts moving through the tree. So this is how we'll actually end up delivering the CYVAV to the trees. And then the seedlings will already have the grafts in them.
Yeah. And another well-known disease worldwide is the banana disease.
Yes.
Is that, if you can say a few words about that, and would you imagine that your solution can address that disease as well?
Yes. So bananas have a number of diseases caused by viruses, caused by bacteria, caused by fungi, and absolutely this can be used. For plants, what it will not be useful for, it really is plants that are grown from seed. So annual seeds. It's not going to be useful for that because it's not going to be possible to get it in there. But if you can grow plants vegetatively, and bananas grown vegetatively, I'm pretty sure, and certainly grapevine is, strawberries are. Citrus trees are graftable, so are apple trees, so are a lot of different trees. As long as you can make a graft or grow something from a cutting, then this is going to be useful. So yes, we would like to go after banana diseases, and a personal favorite of mine is the cacao. So cacao's under huge pressure right now. Probably, my guess is chocolate has about another 10 years as well before the cacao trees are all diseased. And I would not like to be in a world without chocolate. So so we would like to go after that as well. But we're also open. I mean, one of the things that I think Silvec will be able to do is to partner with other researchers and other companies who have an interest in particular trees and particular crops. And they'll tell us, look, we have this terrible problem with this virus. And we will easily be able to swap out the siRNAs for one that will target a particular virus. And then, so it'll be, if this works as a universal platform, it's just going to be unbelievably, I mean, seriously, unbelievably efficacious to what's going on in agriculture. Because right now, trying to develop a virus vector for a particular crop takes many, many years. And most of the time it doesn't work. People don't have the right background knowledge to do it. Yeah. And if you can have a universal vector, then you already know it's going to work. And so that's, I think, where Silvec will place itself. And that's why Raphael is so excited about it. And we're excited too, of course, but he's really excited because he sees all of this potential as a businessman. Yeah.
Well, we're almost at the end of the session. Maybe the last one. When do you expect like a readily commercial solution be in the market? 1 year, 2 years, 3 years? What's your hope?
Yeah, so my guess is it's going to be a few years. And the problem is working with trees. Okay, there's a good reason I never worked with trees. Trees are very complicated. They grow very slowly. And even a very virulent virus like citrus tristeza virus, when you infect it, it takes months before you know if you were successful at infecting it. Months. So everything takes months. And which is why we're doing things in parallel right now. But my guess is that by the end of this year, we'll be able to readily infect citrus. We just had a breakthrough even just a week ago. So we'll be able to readily infect citrus. We'll have tested it in the greenhouse against HLB. What's amazing is, I don't know if everyone knows where Maryland is. We're about 25 minutes from downtown Washington, D.C. on the East Coast. And most people who work on citrus greening are not in Maryland, okay? They're in Florida and California and places like that. But there's one place, there's only frankly 2 places that can work on this disease. One is Florida, because every tree is infected, so they really don't care anymore. And the other place is a half hour away from Silvec at Fort Detrick in Maryland. So we have a place where we can actually work on the disease. And one of our employees actually spent a number of years working on the disease in their BSL-3 greenhouse where you have to shower and you have to change clothes and going in and out. It's just like, it's the same thing for working on SARS. Literally,
Yeah.
SARS coronavirus, you have to do the same thing for working on HLB. So we're going to be able, and they want to partner with us. And so we'll be able to partner with them, and they're only 30 minutes away. So we're in the perfect place to do this as quickly as possible. And we are currently, we are testing the CYVAV, the little thing all by itself, against trees in the field, against viruses. And we are also testing it, about to test it within the next couple of months in the greenhouses at the USDA in Maryland against HLB, just to see HLB. Sorry, that's the acronym for citrus greening. We'll be able to test it against the citrus greening bacteria to see whether or not the normal version of it has any effect. We'll be doing that this year.
Great. And unfortunately, we need to finish. Let me conclude with one comment that we got from the audience, which is the information was shocking and scary, but you gave us hope. So yes. I think it's a good way. It's a good way to conclude. Thank you so much for being with us. It was a pleasure, and we'll be in touch, obviously, because we are partners now.
Yes. Thank you.
So
it was
much.
Delightful being able to talk. I hope one day to meet everybody in person.
We will soon, hopefully. Thank you very much.